Okay, so we're going to start today just because I want to have fun and it's like the second day of class. We've got plenty of time for me to get behind the rest of the course of the class. We will be behind by the end of today. So that's all right. So I take a little more of a we'll make care approach to all of that. So we're going to do whatever come up on here. Those of you that have me in undergrad will maybe appreciate. We're going to start off with something called neuroscience. And I feel like that I need to do a new. Hey, there we go. I'm going to need to update my references. When I started doing this back in like 2009, the Colbert Show was the thing. And so we're going to start off today talking about something that I think is pretty hilarious and very bro-ish called the pump. This is partially driven by, so this, of all the things that I found when I started my faculty position here that was left in my lab was this. And so when one of my PhD students, when Robbie finally mercifully finishes, which I hope will be this year, we'll see, then we're gonna crack this bad boy open and we're gonna actually try some. It's probably, you know, he did his master's here and all those things. We're gonna try this. But it's called,
is it expired? I
have no idea. It's like powder. It was probably expired when I got here.
You're gonna make it try it.
No, we're not. Anybody ever taken this? It's called Nano Vapor. No? Okay. So Nano Vapor is in essence a pre-workout. It's what it's advertised as. But I can pass this around, but it's hilarious because of the things that are written on here. Okay. So it talks about, let me see here, directions. Nano Vapor is extremely powerful, so do yourself a favor. Start off by mixing 1 serving with cold water, downing it about 15 minutes before hitting the weights, then grab some heavy. As you develop respect for and understand the power of Nano Vapor, you can gradually increase the dosage to 3 servings, which is a whole packet, with cold water before working out. So it talks about like, due to the extreme reactivity of the Nano Vapor formula, make sure the packet is stabilized for at least 10 to 15 seconds before opening. Otherwise, vaporized particles invisible to the naked human eye may escape the confines of the packet. So, right? You guys know what that's called? It's called bro marketing. Okay. So I'm losing some of the Nano Vapor. Nano Vapor is a pre-workout. Okay. We'll talk about something called the pump, but there's a thing in here, right? We should ask Skillet if she knows what this is for or not, because it is in some ways pertinent to their lab. So this NO that is right here, anybody know what that might stand for? Anybody taking a pre-workout like this? Gabe, you have your hand up. It is nitric oxide. We will learn about nitric oxide when we talk about the cardiovascular system. One of the things that nitric oxide does very well is it vasodilates arteries and arterioles. Okay. So in theory, you take this and it has this thing that makes your blood vessels dilate. Well, that could be good, right? They're going to dilate. We're going to get more blood going everywhere. You get too much, your blood pressure drops, you pass out, like whatever. Like those are side effects, right? Side effects. Nano Vapor. This is going to let us, then I'm going to go in there, I'm going to grab some, apparently heavy means heavy weights. I don't know, I'm not up on bro. Grab some heavy weights. We're going to do some weights, and then I'm going to get all this blood going to my muscles, and then I'm going to feel like all jacked up, which means I got to pick my shirt up and look at myself in the mirror. All of the bro-ish things. Brady knows. Brady's done it, haven't you? So y'all have at least seen the bros. It's Wednesday. What are the bros lifting today? Chest. I mean, it's always chest day, right? It's like second chest day of the semester. No, it's never leg day. Buys and tries. Buys and tries. It's probably like shoulder day or something. Yeah. Buys and tries. Okay. But this, in theory, then will lead to one of the things that happens when you work out is you do actually increase blood flow to your working muscles, whether you take the Nano Vapor or not. Which is one of the reasons that this thing is probably all marketing and shit, is that you don't need the extra nitric oxide to get more blood flow to your muscles. And even if you did, having that extra blood flow is not going to do anything for you in those particular ways, right? So you go and you get all pumped up. You're like, oh, I'm huge. And we put on my medium shirt and go to the bar. And then 2 hours later, they're back to their normal size. They've shrunk back down to normal, right? So the pump that the bros refer to is not, I got in a good workout. It is not even like a thing. There's a thing called acute hypertrophy. You work out, you increase the hydrostatic and the oncotic pressure. It drives plasma or intracellular fluid into the cells, into your muscle cells. They swell. Therefore, you get this acute increase in actual muscle size that we can measure. But then in a couple of hours, hydrostatic pressure goes back down, oncotic pressure, after we get all of our metabolites back where they're supposed to be, goes back to normal. Guess where the water goes? Back out of your muscles, and you inflate yourself back down to your regular size, whether you've had nano vapor or not. There's a whole slew of things in here. So it talks about that it has psychoactive pre-workout matrix. You guys know what a psychoactive pre-workout matrix is? There's another name for that psychoactive pre-workout matrix. Caffeine. Okay? It has caffeine. It won't say how much caffeine is in there because it's a proprietary blend of something. It's just caffeine. That's all it is. Ignites white hot intensity and explosive strength. Again, caffeine. Anabolic factors force extreme muscle gains. There are no anabolic factors in this. None. Not a one. It has creatine in it. Creatine is not an anabolic factor. Might have some protein. I don't know. There's no, I mean, maybe it has testosterone. You know, but I mean, it literally says, do not shake the pouch like it's going to explode or something. We've shook the pouch. Nothing happens. So anyway, be wary. This is bro science. You take the pre-workout. That's fine. It has things in it like creatine and some other things that theoretically there is some scientific support that they are beneficial. There's probably not enough creatine in there. Taking it once before a workout doesn't do anything for you in that matter. All that's in there, probably that works, is caffeine. Go to Walmart for $4, right, Brady? For $4, you can buy 200 caffeine pills or 100 200 milligram caffeine pills for $4. This whole pouch is probably like $6 or something, right? You can look it up. You can Google. They still make this at what that pill costs, whatever. There's caffeine in there. Let me see if there's anything else that might actually be. I mean, there's this huge list of things, right? And I'm actually not even sure that it doesn't even have nitric oxide in it. It has L-arginine, which is a precursor for nitric oxide. It probably has beta-alanine because you need to feel like you're going to be itchy. There's a little bit of support that maybe beta-alanine does, in fact, actually increase exercise performance. Yeah, it has beta-alanine in there. Of course it does. Anabolic, anti-catabolic signaling, that is not what beta-alanine does. It only has 117 milligrams of caffeine. So it's got barely more than an 8-ounce Red Bull. Per servings, there's probably 3 servings in there. So there's 350-ish in the whole thing, right? Go get a grande latte at Starbucks and you're going to get more caffeine than that. You're fine. Anyway. So, bro science, do we need a pre-workout? The caffeine helps. It really does. The caffeine does actually help. There's plenty of scientific support for that. Everything else in there, I don't know. Probably not. Okay. Christine, do you have a question?
No, I was just going to say that was like $30.
So the tub is like $30. Okay. But it might explode, so they won't even send it to us, right? Like if I can't, UPS won't handle it. Anyway, we found that in my lab and we thought it was pretty hilarious. And so I can bring it in. Okay. Now, can you guys envision a scenario whereby on your first test, once we've talked about how muscles actually work and contract, or how the nervous system communicates with your muscles to make them contract, how we control force production, Or what happens about how we get blood flow to move around when we need it to during exercise? Maybe a critical thinking question could be, I could give you something like this and say, talk to me about whether or not you think this could be effective and why, right? Here are the things that are in there. Here's what they say that they do. If that's real, could it work or could it not work? Defend your answer, okay? That's the level of stuff that I'm wanting us in some ways as, remember our discussion about being a graduate student, right? Even although sometimes we have graduate student bros that are still like, I'm taking my pre-workout, fine, okay? Could we actually try to explain that, okay? And what the supplement companies will do in a lot of instances, because there's a huge amount of money to be made, billions in supplements, is some of the things in there are real, or they sound real. They do some things that are related to functioning muscles or the cardiovascular system, but like they don't talk about that if you just exercise, if you can't vasodilate your things correctly with or without that L-arginine, like you can't exercise. It's not a problem. Getting more blood flow after you work out to a muscle, that's not the limiting factor to increasing muscle size or anything else. Okay. We're going to start off kind of class talking about muscles. I was thinking about this before class today, and I probably need to go in and I start off by doing kind of some basic neuromuscular anatomy. Okay. Then we do some central nervous system anatomy and kind of how those things are connected and the signaling between those two. In theory, what I should probably do is we should start off with the nervous system side of things, because that's where all muscle contractions are going to be initiated in the nervous system. Okay? Not all, the vast majority. Okay? I should probably flip those two things around, but say loudly. Okay? So here's what we're going to try to do. This is the overarching goal, and I will try to do this with every lecture, with every kind of set of things that we're going to do, is here's the point we're trying to get to at the beginning, and then we'll kind of work backwards from there. Okay? So if I want someone to do something, let me look around. Can I get a volunteer? Anybody want to do something for me? Anything? Anybody? Okay, Brady. Brady, stand up. Brady's tall. That's not going to work very well. Okay. Brady, can you go down and like do a squat and hold a squat? Your gene's going to be okay if we do that? Okay. So Brady's going to do a squat. Brady, have you ever done a squat before?
These jeans on.
Okay, you wore your skinny jeans today. Okay, that's fine. Pandemic 15. I'm right there with you. All right, Brady's doing this. All right, Brady, stand up. Go back down. Stand up. Touch your nose. Okay. All right, sit down. Sarah, how did he do that? Very good. His nervous system sends signals to his muscles. His muscles generate force. The muscles have their force. That force is transmitted along connective tissue to tendons that are then going to pull on bones, right? Bones are going to sort of articulate around a joint. And if the force is enough to overcome gravity or whatever it is that his leg weighs or something, or what Brady weighs, then we can see the output as movement. Okay, so the big picture on all of this is we are trying to go from the idea of the brain all the way down to the muscle that is ideally then going to give us force, which results in movement. Okay? Where in the brain would that begin? What part of Brady's brain is going to, that's a loaded question. So what's the major part of Brady's brain that's going to play a role in initiating this? And like I say, Brady, do a squat. He may or may not really think about it, but it's sort of thinking, do the squat. Okay, squat, and I'm up, touch my nose, sit back down. Where does that start? Okay, motor cortex. Very good. All right, so We could talk about it being the primary motor cortex, right? And there's going to be input from some other areas that are going to move around. But for the undergrads, I call this cognition. You're thinking about doing something. And we're going to trace our way from thinking about doing it in the brain, cognition, all the way down to what they're going to think of as an actual contraction that gives us force and movement. Okay? There's a series of steps and a series of control points that are going to move us all the way down through all of this. Okay? Those steps are important. Those control points are important because if one of them gets messed up, all bets are off. Okay? Nothing works, or it doesn't work in the way that we want. Okay? Additionally, if I had Brady just keep doing squats over and over and over again, at some point he would fatigue. At some point he'd have to stop. And that, his brain, he might be trying to do things, but we can't get to this point down here for a host of reasons that we will talk about later in class. Okay? That's how we're going to kind of get at with our first 3 or 4 kind of lectures. Okay? Is what are these steps going to be as we move through here? What are the primary structures? What are the primary signaling molecules? What's the primary sort of anatomy and physiology that is at play here? And once we learn some of that, we're going to put it all together and talk about, well, how did Brady know how much force to generate to allow himself to squat but not jump, or to sink down and stop at that particular point, okay? Or to touch his nose but not punch himself in the nose, right? You guys ever try to pick up something you thought was going to be heavy and it wasn't? You like flop over backwards, right? Your brain made an estimate of how much force it needed to generate, and it was wrong, okay? And then as you go backwards, you realize that you're off balance and you got to make some estimations about how to get yourself back on balance. The brain's really good at these things, but we'll talk some about what is going on, how does it actually do that, okay? Okay, so I'm going to go pretty quickly through a fair amount of this because I'm hopeful that for the basics of the anatomy and the kind of basics of the physiology that you guys have seen a lot of this before, I want us to focus much more so on kind of process and putting it all together. Okay, so I'm going to skate through a few of these things. If there's something you're not sure about, please ask, and I'm happy to spend more time talking about it. Okay, so we've done this. This is kind of also going to be another piece that goes into kind of force and movement with muscles, is that structure imparts function. Okay, structure imparts function. You have different muscles that have different shapes, right? And you have different lengths of tendons, and they attach to bones in different places. You have motor neurons in your spinal cord that are in different places. Their structures are important, okay? And so muscles generate force, which allows us to move. We have motor neurons in the spinal cord that are going to be connected sort of downstream of the motor cortex. These are going to be part of the things from the nervous system that are going to provide a signal to the muscle to let us contract, okay? This idea of thinking about doing something and then doing it is oftentimes called voluntary control, okay? The only times when you don't have voluntary control over a muscle contraction is if you get a cramp, okay? You guys ever had a cramp? Right? Not voluntary control. Do not want my muscle to contract, and it is. Or if you come to my lab, we'll slap some electrodes on you and we'll zap you with electricity and we'll make them contract artificially, either by stimulating the muscle or we'll stimulate the motor axon or something if we want to do all of that. Okay? So, but there's a signal that says, hey, muscle, do this, right? So we're communicating in this particular way. Okay.
Also, whenever you get that twitch, another workout, you get a twitch.
Huh?
It's not really a cramp.
I would know it's probably not voluntary. So you probably have a little bit of calcium disruption somewhere that's doing all of that. So are you usually a little bit sore when that happens? Or are you just like, no, it's just kind of like randomly twitching? It's fine. Okay. So The other big thing, because all of this is voluntary, because we have a way to communicate from the brain to the muscles, what it lets us do, okay, it is one of the things. Anybody ever seen is it like the Siemens robot or whatever, the MIT, the dog robot that can like stand up and climb upstairs? Like they're going to be Terminators at some point. They're all coming for us, right?
I had them do an agility course. Like,
yeah, I know, it's crazy. And then now, of course, the Tesla guy, whose name I can't remember, they want to make a freaking robot or whatever, because of course they do. But one of the things that's hard to duplicate when you use artificial intelligence or computer-driven things is that movement requires coordinated contractions between multiple muscles across multiple joints, okay, and the precise control of the force production from every one of those muscles, okay? You have to do just the right amount of force at just the exact right moment just to walk. And if you think that's not true, come watch my daughter try to run around sometimes. Watch a toddler learn, or an infant learn how to walk or crawl, okay? Or I guess we could go to the Mont and get you 3 swirls and then see how things go, okay? This is difficult. And we have this really, really cool ability to very precisely manipulate and manage what muscle is on and how much force that muscle is generating at any given kind of instant in time, okay? And so if you're really interested in that kind of stuff, you should take the doctoral level neuromuscular physiology class with Dr. Pereira in the spring. But there's a whole bunch of other things that are going on, both in the motor cortex at the level of the spinal cord, that we don't really have time to get into about how we do some of this, okay? But that's the cool thing about this, all right? That's the cool thing. It's very difficult. I can artificially make 1 or 2 muscles contract in the lab, but it's very hard for me to make them work in concert to allow you to like ride a bike or walk or something like that, because Lord knows we tried. In my PhD, we worked with people with spinal cord injuries. They could not contract, for the most part, their muscles of their lower body. And we tried to get them to do things to try to ride a bike. And you have to stem the quads and the glutes and the hamstrings and the TA and the gastroc and maybe the soleus, kind of all simultaneously at just the precise moment to get what you want. Okay, you wouldn't stem the hip flexors. That was a problem. And it's hard because then they would all fatigue at different rates, and it's a mess, okay? So in this way, we are extraordinary. Our system is extraordinary. So you've got more than 600 muscles in the body, okay? Think of muscles as Russian nesting dolls, okay? You guys know what a Russian nesting doll is? Or you're nodding your head, but what's a Russian nesting doll? Exactly. And they're all identical, right? You open it up, oh, there's another one, and another one, and another one. Okay. Muscles are, in some ways, a collection of tubes that are like Russian nesting dolls, okay? You've got a whole muscle. That whole muscle is made up of a thing called a fascicle, right? It looks like a muscle, but it's just, kind of a bundle. You open those up, you open the fascicle up, and there's more tubes that are muscle fibers. You open a muscle fiber up, and there's all these myofibrils. They're all just getting smaller and smaller in this way, okay? So muscle cells are called muscle fibers. And generally, within a given muscle in your body, those fibers are all going to run parallel to each other and run in the same direction, okay? So in my bicep, all the fibers are running kind of along the axis of pull for the most part, okay? There are some exceptions to that. Each individual muscle fiber or muscle cell, this is the thing that makes it different than a lot of other cells in your body, has many nuclei. Muscle cells are giant, okay, in the world of cells. You can see them with the naked eye. If you let me cut open your arm, we could tease out individual muscle fibers, okay? They might be a couple of centimeters long. You can see them, okay? Because they're so big, they need multiple nuclei to be able to supply all of the protein and structures and things that go in there, okay? Also, strangely, their nuclei are not located in the middle of the cell because that would occupy space that they need for actin and myosin and things to allow that muscle fiber to contract. So their nuclei are off on the periphery, which also makes them different from a lot of other cells. Okay? So muscle is approximately 75% water, 20% protein, and about 5% salt and some other substances, so some electrolytes. Okay? Much like you as a person, muscles are mostly water, hence the reason we can get a pump afterwards, get more water in, they look bigger, dehydrate yourself, and the water goes out and you look a little bit smaller. Think about the bodybuilders, right? If anybody has ever done those kinds of things, done some sort of bodybuilding competition or fitness competition, they take salt pills, they dehydrate themselves, it drives the water out of their muscles. That's why they all look like they're a football or something. Their muscles are real dry in that. Okay, so here's our Russian nesting doll kind of set up here. You've got a whole muscle. That muscle is going to be surrounded by some fascia, so it's surrounded by connective tissue. In the muscle, there's all these bundles of muscle fibers that are called fascicles. Each fascicle or each bundle is also going to be surrounded by some connective tissue. Open up those, and you've got these individual fibers that are going to be in there. They've got connective tissue around them. Each fiber has these myofibrils. So this is one fiber. They've got myofibrils inside of them. And then the myofibrils have these protein filaments that make up. So there's this kind of nesting structure that's going to go on. We're going to spend most of our time talking about what happens at the fiber level, okay? And then what happens down with the proteins and the filaments that are going to make up our individual sarcomeres. So we'll talk about some kind of overall structures that are in the fiber and then really what's going on here. Because to be honest, it's the proteins and the individual actin and myosin molecules that make up a muscle that are formed together into these thick and thin filaments. That's really where all of the action in the muscle takes place, okay? So it's the proteins interacting with each other that are what generates force, and that's what generates movement. But we need some of the things at the fiber level to provide us energy or to help us signal and tell these things what to do. But really, so we're going to go here and kind of down here. Okay? So I mentioned that there's connective tissue that surrounds all of these different things. The outer layer is called the epimysium. The one around this, we've got the paramysium and we've got the endomysium. I don't really care that much about this. The really important thing about connective tissue is because there is connective tissue, muscles are stretchy, okay, which is good. And it lets them transmit force from one place, okay? I can contract one little section of a muscle, and because of all of this connective tissue, it should be able to transmit some of the force from there along the entirety of that muscle, okay? That way, you don't have to engage all of the muscle all the time in order to be able to get movement, which will become very important later. Okay? So we need it for scaffolding. It's a conduit for some blood vessels and nerves. Again, if you take neuromuscular physiology, we'll talk a little bit about kind of nerve regrowth and nerve regeneration and things if you've got damage out in the muscles, and that's when the connective tissue comes. Okay. All right. So a few quick things about structure. Let me check and see if Drew is saying something about the cortex. Okay. Relate to all of that. This is a cartoon cross-section of a muscle fiber. Okay. Cartoon cross-section of a fiber. So we've taken one, you've got a little section of it, we've cut it open. So a fiber is a cell. Like all cells, it has a cell membrane. It looks just like most other eukaryotic cell membranes or other cells in our body. It's a lipid bilayer. It's got protein through all of it. It's got a little bit of connective tissue. It's got a basement membrane. It has all these other things in there that make it look like all of our other cells and keeps it healthy. That cell membrane is called a sarcolemma. Sarco is either Greek or Latin, and I cannot remember which, but it basically means muscle. Okay, sarco means muscle. So there's going to be some basement membrane around there that we'll talk about. Coming off of the sarcolemma are going to be tubes. They're called transverse tubules. And it's a tube or a channel, however you want to kind of make sense of it, but you're going along and there's a hole in the sarcolemma. And that hole leads down into the internal structures in that muscle fiber. Okay? That tube is going to have essentially be the same structure as the sarcolemma, which means it's an excitable membrane. It can transmit an action potential, which is the real purpose of these transverse tubules. Pardon?
Are they gated?
So the tubule itself out here at the sarcolemma is not gated. You've got to get a sufficient amount of charge. So the tubules are going to lay inside the cell next to the sarcoplasmic reticulum, which is this kind of red, webby looking structure that's in here. And it is just a storage site. It's basically a closet for calcium. Okay? And it's going to go all over the inside here. The T-tubules are going to lie in parallel, kind of right next to what's called the terminal cisternae on the sarcoplasmic reticulum. And there is a gate there. Okay? And so you have to have a sufficient action potential or sufficient change in membrane potential to move down the T-tubule to interact here to tell the sarcoplasmic reticulum to open up and release its calcium.
That's for the
So there are sodium and potassium pumps on the sarcolemma and in the T-tubule.
okay.
And because of the nature of the structure of the T-tubule, we'll get into this much later in class, but you sometimes have a hard time repolarizing the T-tubule and it leads to fatigue. So you cannot generate a sufficient amount of force because the area is so small that if it contracts over and over again, it has a hard time, it doesn't have enough sodium and potassium pumps to repolarize as quickly as the sarcolemma. So you can get some problems in there, which we can actually experimentally measure and figure out is the fatigue from this or from something else. Okay. Those are good questions. There's also mitochondria that are going to be in there. We'll save some of our talk on mitochondria when we get to metabolism, but inside the fibers, you're going to have a host of these things. The amount that are in muscle fibers is going to depend upon both the muscle fiber type, okay, if it's a fast twitch or a slow twitch muscle, and it's also going to depend upon how well aerobically or endurance trained you are. This is one of the things that's plastic in those muscles that can go up or down based upon activity. Okay? As I said, the nuclei are out here on the outside, and then muscles also have some really cool things called satellite cells. Satellite cells are muscle stem cells. Okay? Does anybody remember? I'm sure some of you do. My biomedical engineers probably do. What stem cells do? What's cool about stem cells? Repair, yes. It can become anything, right? Satellite cells are not pluripotent stem cells. They can't become anything. They've moved forward and arrested in the development. They can only become muscle fibers. But under a microscope, it's hard for us to tell the nuclei from satellite cells. But what they do is, Gabe is 100% right that when we exercise, when we resistance train, or if you damage your muscles, satellite cells play a really, really key role in repairing damaged muscle fibers, and they play a really key role in growing them and making them bigger as you go forward. Okay? There's a special anabolic hormone called insulin-like growth factor 1 that is particularly potent at activating satellite cells. We'll get into some of that later on when we talk about resistance training. Okay, but they're in there and they are important and they're unique to skeletons. Let me go. Here. There we go. Okay. So I, for the most part, talked about most of the important things on the sarcolemma. The thing that's really important about the sarcolemma, as we're going to get into our signaling here, is that the cell membrane of muscle fibers is excitable. Okay. And what that means is that it can generate and move or propagate an action potential. So in that way, the sarcolemma is a lot like an axon or nervous system tissue. Okay? So we're going to use that property to move electrical signals along it very fast. Electricity is good because it goes fast. Okay? It also has a whole host of receptors on it that are going to bind hormones and other sorts of things to kind of tell the muscle what to do. Okay? I mentioned that there is a basement membrane that's going to help to kind of fuse the epimysium, or the outside connective tissue, and attach muscle fibers to the neuromuscular junction and tendons. Okay, it's going to also, in the basement membrane, it's kind of on the outside of the muscle fiber. It's going to hold acetylcholinesterase, which is an enzyme which helps to terminate the synaptic transmission. It's going to go on there. The membrane provides scaffolding so when we activate our satellite cells, they kind of know where they're supposed to go in that way. And it also helps to help kind of regulate the neuromuscular junction. A lot of interesting stuff being done now. Research on the basement membrane, it's a lot more important than we used to think that it was. That always just kind of there and it holds things, but it seems to play some signaling roles. It seems to play some additional regulatory roles that are going to, again, it's kind of beyond the scope of what we try to do in class. So this is just showing you here, right? Like here's the sarcolemma, here's the extracellular matrix, and kind of the basement membrane stuff that's going on out there. One of the things that you guys might have heard of that's going to be in the basement membrane is a protein called dystrophin. You have muscular dystrophy, you lack this dystrophin, or you have a misformed dystrophin, depending upon kind of which flavor of muscular dystrophy you have. So that means you can't anchor these muscle fibers correctly, and they become very weak. They become very easily damaged and disrupted. Okay. We talked about T-tubules. We talked about sarcoplasmic reticulum. That's fine. Okay. To get to Christina's question, okay, at this junction between the T-tubules and the sarcoplasmic reticulum, we're going to be able to have a signal, okay? So we've got T-tubule here, right? It is also a lipid bilayer, just like the sarcolemma. And at various points along that membrane, there are going to be these receptors, which are called Dhp receptors. It stands for dihydroperiod, okay? It is a voltage-gated or a voltage-sensitive receptor. It needs an action potential to come by and turn it on, in essence. Right? So when DHP senses an action potential coming through there, it is going to signal to a receptor that is on the surface of the sarcoplasmic reticulum that is immediately adjacent to it here. That receptor is called the Rhinidine receptor, or the RYR receptor. Okay? The Rhinidine receptor has multiple little subparts to it, but under the microscope, it looks like a sink drain with a plug in it, okay? And when the DHP receptor gets activated by an action potential, it's going to cause a conformational shift in the Rhinidine receptor that pulls the plug out of the drain and lets the calcium flow down its concentration gradients out of the sarcoplasmic reticulum and out here into the cytoplasm of the muscle cell, which will become very important very soon. Okay. Does that make sense? Because this is, if this gets messed up, and it will, then we cannot get muscle contractions. Okay? Cannot get muscle contractions. So we're going to talk a little bit later on, probably not today, but next week, this process of going from an action potential in the sarcolemma into the T-tubule to get calcium release is a process called excitation contraction coupling. Right? We need the calcium to get force production. The other problem, though, is if I just inject calcium into your muscles, I can make them contract, but then it will also activate enzymes called calpains that will tell those muscle cells to undergo apoptosis and kill themselves. So, having extra calcium floating around inside of a muscle cell for very long leads to cell death. So we don't want that. So we have to have a way, once it's gotten out, to quickly and efficiently get it back in so that we can initiate the muscle contraction, but not initiate programmed cell death. Okay? And so what we have a pump that is here. It's an enzyme. It's called the calcium ATPase, the calcium ATPase pump. And it is oftentimes referred to as Sarcome, which is just meant to be sarcoplasmic, something, right, calcium transport. Basically what Sarca does is, Sarca is like a vacuum cleaner. It sucks up all the calcium that's out here in the cytoplasm that's been released, and it moves it back into the sarcoplasmic reticulum. Okay? It's very efficient. It does it very well, but it uses energy. Okay? It uses energy. There are some estimates that repolarizing the T tubules and the sarcolemma and pumping calcium back in accounts for like a third of what your resting metabolic rate is. It's a huge amount of energy that we expend every day doing all of this. Okay? We'll talk a lot more about Sarca function because it differs between individual muscle fiber types, and things that can happen with Sarca as well as with Rhinidine can affect how much force you can generate, how long you can generate that force, and can tell us some kind of interesting things. Okay. Do a little quick intro to mitochondria. Skillet, which it's hard for me to get used to that, but yes.
Sorry, I have a real quick question.
Of course.
You know why the cell undergoes apoptosis?
So the thought is that what generally that is an indicator of is that there's damage to the sarcoplasmic reticulum. And if the sarcoplasmic reticulum is damaged and you're leaking calcium out, then that's not good. And it's better to just kill the cell off than to try to repair the sarcoplasmic reticulum and those kinds of things. Just, you do not want calcium where it's not supposed to be at all costs. So one of the things that happens, anybody ever been sore? Like you work out, you've been sore? You are sore because of inflammation. If you say you're sore because of lactic acid, I'm coming after you with the scissors, okay? But you're not sore because of lactic acid. You're sore because you damaged your muscles. You had little tears in the muscles. And part of the things that were associated with that, very likely were going to be that you tore the muscle fibers, and you tore the sarcoplasmic reticulum, and you released the calcium where it didn't need to be. You've got some small population of fibers that have killed themselves. When your muscles are damaged, you're sore, you're also not as strong as you would normally be. It's not because you're sore. It's because you have some fibers that do not function. Those fibers are basically dead. And so it does that, it cleans them out, the satellite cells get turned on, you replace them, and then within a week or 10 days or 2 weeks, you're pretty much back to normal. We know this because we can give you a drug or a supplement that will blunt the effect of those calpain enzymes that the high calcium levels activate, and you don't lose as much force and kind of recover from damage faster. So that's not what you want. You don't want that. You need to clean out those kind of damage fibers. Christine.
So satellite cells,
Yep.
are they the ones that, not necessarily determined, but are they the ones that could possibly switch or influence what type of muscle fibers you have after working out? So influence more mitochondrial makeup or growth within the muscle cells?
They certainly play some influence on growth.
Okay.
That's for sure. Because we can, or they have, and they do this a lot in animal models because it's probably unethical in humans to do this, but they will irradiate the muscles and essentially kill off the satellite cells. And they have those animals work out, or they treat them with testosterone or something that should activate and cause hypertrophy. And they get a little bit, but not very much.
Not as much as the healthy muscle cell.
Okay. So we know that they influence that. The role of satellite cells and getting more mitochondria or shifting mitochondrial function, I don't think they're particularly important in that. And then the change in fiber type that we will talk about, it probably doesn't have a lot to do with that, but I can't say that 100% for certain. Weirdly enough, the shift in fiber type we actually think is driven by the motor neurons and the nervous system connection to the muscles. you guys will see. They've done some fun experiments, especially they do it in animals. One of my buddies in his postdoc, they cut nerves and then they swap them around. And so you can take a nerve, cut it that was going to fast twitch muscle fibers, and you can connect it to slow twitch fibers, and then take the one from slow twitch fibers, and swap them around, and the fibers will then switch in that particular way. So we think there's something about that connection seems to drive it more so than kind of the satellite cell side of things. Even though what's changing is actually in the muscle. It's going to be part of a piece on the myosin, part of the myosin. So it's a good question. Great.
What age does muscle carry?
I'm sorry?
So what age does muscle carry? When does it slow down? As far as those.
I don't have a good answer for that, Brady. I mean, we see, so the rate of repair is interesting. So we know that older muscle is more susceptible to damage than younger muscle, but a lot of that may be driven simply by less activity, less use. We can see hypertrophy, and again, this is, we'll talk about it now, we'll get into it more so when we do resistance training. Up until about the age of 80 or so, you can take people and have them do resistance training, and they can see hypertrophy from wherever they were starting. After the age of about 80, give or take, then they don't lose muscle like most aging kind of models do. You can stop the loss of muscle, so you can stop sarcopenia, but they cease to be able to hypertrophy. Which it doesn't make a lot of sense to me that we're stopping one thing, but you can't kind of go ahead and increase. So maybe around then, is that due to some sort of misfunction in the satellite cells, or an inability when you use the satellite cell to repair something to then get another satellite cell into that muscle? I don't know. I would need to look some of that stuff up. So I'm not a super expert on the satellite cell. But the repair, it's complicated. So as I told you guys on Monday, the answer is always it depends. So, okay. Other questions before I move on? No? Okay. Yay, here's the mitochondria. We all have bacteria in our muscles, right? Mitochondria have their own set of DNA. There is a sort of symbiotic relationship. We at some point put these things in. They look like this. If you're curious about mitochondrial DNA, right, Gabe? So Gabe can tell you all about it, or the mitochondrial genetics of the mitochondrial DNA. So that's one of the things that our new faculty member, Dr. Ellers, is interested in studying. It's not just mitochondrial function, but the genetics of mitochondria and what happens with all of those things. But you have them. They're like little bacteria that are going to be inside your muscle cells. This is a place where we use them to make ATP via aerobic metabolism, okay? Using the Krebs cycle and oxidative phosphorylation. We'll talk about more of that than you probably want to know, unless you've already had biochemistry or something in a few weeks, okay? But they're there, they're in the muscles. Their numbers are going to vary based upon fiber type. Their numbers are going to vary based upon. We'll come back. We'll talk about the inner and the outer membranes and what's going to be in which direction stuff is going to move as we work our way through where the Krebs cycle takes place and where the electron transport chain is located and how we get ATP in and out and things can go through. Matt?
Are the mitochondria genetically identical or are they different than
So, okay. My assumption is that all of your mitochondria are genetically identical, but your mitochondria and my mitochondria are not genetically
So
identical.
99.9%.
Like your whole body?
Like your nucleotide, so it's like 4 letters. Those 4 letters are identical 99.9%. And then there's each mitochondria, there's like 2 to 10 copies of DNA, and that can vary. So it depends. So even your own mitochondria may have different DNA. And so if you compare it to another person, the chance of your own being different, but not that much. Okay.
There's people with like mitochondrial disease, or like there's things like mitochondrial transplants. Like,
So it's funny that you say that, Ray. I don't know that we can do it yet, but I had a graduate student in my very first job, like almost 10 years ago. That was his. He was going to start a biotech company. He wanted to, and they were going to genetically engineer mitochondria and then inject them into people, so you could all of a sudden be great endurance athletes. So we even named our intramural softball team after it.
What was it?
The Mito Loaders is what he wanted to call it. I'm like, okay. I mean, you guys have seen what Pfizer named their vaccine. It's the dumbest name ever. So we've been talking about satellite cells. We've talked about nuclei. Here is, I think that's an H&E stain. But what you can see here is these little things that look like maggots or little pieces of rice. This is a muscle fiber. It's purple. And all of the things that you can see in white are either myonuclei or their satellite cells. Okay, so you've got a huge amount of them that are going to be in there. The most likely thing is that some of these things here are going to be satellite cells. Satellite cells tend to have like a double membrane around them. The nuclei only are going to be up kind of next to the sarcolemma.
So is there a relationship between the amount of nuclei and the type of muscle fiber?
No.
Okay.
Not that I'm aware of. Not that I'm aware of. So there is some thought that the number of nuclei that you have may in some ways be related to your potential to increase muscle size, right? There's a reason that bacteria are not like as big as this room, right? There's a surface area to volume to whatever protein they can make. So in theory, if you start off with a bunch of small muscle fibers and you have a lot of myonuclei, you have the potential to supply stuff to bigger and bigger fibers. And if I have more nuclei than you, maybe that could account for some of the genetic variation in those things. But it's incredibly difficult for us to study because I got to get a biopsy. I got to go all the way and look down at this. I'm looking at one small little piece, and I got to try to get from a close location, but not the same location before and after you've done training. And it's a mess in some ways, but it's possible. So this is just showing, this is kind of some fun staining where they've got these satellite cells. Here's one that's getting activated. So this muscle has probably had some damage that's going on. We've activated this. And once it gets activated, it's going to advance in its cell cycle. And what it does is it begins to divide and it splits into two. And it keeps one of them that's going to be the new satellite cell, and the rest are going to work their way down through. No, why can I not think today? I should say that should be mitosis, but we just have one cell. But depending upon how many fibers have been damaged, you can go through sort of mitosis and work your way into this. And they make things that are called myotubes, and these myotubes all fuse together, and then they begin to eventually get to the muscle fiber. If this stuff is interesting to you, there's a huge amount of research and kind of interesting cell signaling stuff that goes on that controls a lot of these different things and how exercise can activate them or how different drugs and things can interact in the midst of all of this to do things. Okay? All right, so we talked about that. Next thing I want to do with my time is we spent time talking about fibers. Now we're going to talk about kind of the protein level things. Okay, so remember, a fiber is made up of many of these little kind of mini fibers that are called myofibrils. And the myofibrils are made up of these individual sarcomeres, and the sarcomeres are made up of filaments that are these actin and myosin proteins that look like this. Okay, so it's these two proteins and their kind of individual arrangement into filaments. Actin molecules are little balls or little spheres, and they get stacked end to end and they wind around each other. and that becomes kind of rope-like structure. That's going to be an actin filament. And then myosin molecules they look kind of like sperm, to be honest. They have a head region and these tail regions. And then you're going to get all the tail regions that end up getting hooked together, and the heads project off it at different intervals. And so these are called, actin filaments are called thin filaments, myosin filaments are called thick filaments. When you look at them under a microscope, even just under a light microscope, I'll show you guys some pictures of sarcomeres in a minute, it becomes very obvious, right? Actin has a smaller molecular weight. They look thinner. They look less dark and less dense in that way. And the myosin are bigger and they're heavier, hence the reason we call them filaments. Myosin makes up roughly 2/3 of the protein in your muscle. Okay, roughly 2/3 of the protein in your muscle. Myosin is the really important thing. Okay, myosin is what we call a molecular motor. It's a motor. Okay, all movement that you initiate is done by the movement of these myosin heads. Okay, myosin heads will attach to actin and they will tilt or change their conformational shape, and that generates force and it pulls these actin molecules, and that's what generates force. So this interaction is really what we're trying to get all of the signaling down to be able to control. Okay, so we'll talk a little bit about the arrangement of these two things together in sarcomeres, how sarcomeres are arranged into myofibrils, so you can understand how we maximize our ability in muscles to put all this together. Okay. Okay. Here's a sarcomere, right? If you read any exercise physiology or neuromuscular physiology or medical physiology textbook, they will probably tell you that the sarcomere is the basic functional unit of a muscle cell. Okay? Functional unit is not a fiber, it's not a myofibril, it's a sarcomere. Okay? So one sarcomere is going to run from this border here, which we call a Z line or a Z disc, over to the other Z disc. Okay? In a myofibril, you're going to have a sarcomere here, and a sarcomere on this side, so on and so on. And you're going to have this big tube, okay, that is just sarcomeres in series put next to each other running along like this. You've got one myofibril, and there's a myofibril next to it, and above it, and below it, and those are what are going to get circled in to make our muscle fiber. Okay? So the movement that happens when actin and myosin interact is going to happen here at the level of the sarcomere. And what's going to happen is you have this very, very kind of strict arrangement of these alternating bands of thick myosin filaments and thin actin filaments. Then you're going to have this region where they're going to overlap with each other. Okay? We're going to have regions where they don't overlap with each other. I don't really care if you know what the M line is or the H zone or anything else that goes on. I assume in our undergrad class, Dr. Mike made you guys learn all of that or something. I don't really care. You can get a textbook out and you can look it up. The important thing is for movement to happen, sarcomeres do this, and they smush together in the middle. Okay? They're going to pull, the myosin heads are going to pull these actin filaments from one side in towards the middle. When you do a bicep curl, what is happening is there are thousands and thousands and thousands of sarcomeres in my bicep that are all smushing together. Okay? And that generates force. As they pull in together, then that generates that movement that allows me to curl up and have my muscles short. Okay? Don't ask me right now what happens when I lay something when I go back down the other direction. Okay? I'm not ready for that yet. Okay? That's advanced exercise physiology. Sure, we'll talk about it, but it's not for right now. Okay? So that's what's going to happen. All right. That's how sarcomeres work. They can only pull so far, and they will butt up against each other. Okay? Or if you do something weird, like go to yoga too much, and you stretch them out too far, you can actually pull the actin filaments all the way out. There's no longer going to be any overlap. That's not what happens in yoga. I just hate stretching, so I like to make jokes about people that do it. Okay, they're mainly directed at my wife, but that's another thing. Yes, ma'am.
So with hypertrophy, when we're increasing our muscle, are we also increasing the amount of sarcomeres within our muscle as well?
Yes, absolutely you are. So in a fiber, there's going to be, actually I'll draw. Don't laugh. I'm not an artist. Okay? What is that? It's a muscle fiber. Okay? So it's a cross section. This is a fiber. Okay? In that fiber are going to be all of these myofibers. Okay? Each of them then has, if you pull one of those out, then they're going to have these sarcomeres. Okay? To make my muscle bigger, the fiber is going to get bigger. So now I have more and more and more and more. I'm just getting lazy at drawing. Okay? I have more and more individual myofibrils in here. Okay? Okay, next question maybe got caught. Did myofibrils actually get bigger? I don't think so. But I can look that up.
Yeah.
I think what going to happen is you get new myofibrils.
Okay.
You get new myofibrils. And so as I get more of those, then the fiber itself gets bigger. And as your fibers get bigger, then your whole muscle begins to get bigger.
Okay.
Okay. So what happens is here's multiple sarcomeres, right? Here to here, basically. There's going to be a sarcomere. Then another one and another one. See how nice and orderly this looks like? Okay. It also creates a pattern, a striping pattern of these alternating filaments, which is why skeletal muscle is said to be striated. Okay. This is just, under a light microscope. It's going to look this way. Right? So when these things are going to contract, every one of these between these two areas in here are going to squeeze in towards the middle. They're going to start squeezing all of them. Okay? And because out here at the Z line, they're going to be anchored to connective tissue. As they move, they pull on the adjacent sarcomere, and so on and so on and so on. And therefore, eventually, we can transmit some of that force up to the tendon, and we can get it. Okay? If you care about the different bands and discs and all of those things, here it is nicely laid out for you. If that's a thing that appeals to you, I assure you on the test, I'm not going to ask you to explain to me what the I band is and why it's important or how the I band changes during concentric muscle contractions or something else. It's important, it will change, but we're going to talk about some other details that I think are probably. Okay. It's important also to understand in the myofibril the three-dimensional arrangement of myosin and actin filaments to each other. Okay. So what you guys can see here is we've taken a myofibril, it's running this direction, and we've turned it so you're looking down into the end, okay, that cross section of the fiber, or of the myofibril. But what you will note is every pink one of these things is a thick filament, and every blue one is a thin filament. So the myosin heads are going to project off of their central kind of filament structure at these kind of precise angles. They're going to make a hexagon. Okay. And I should think about that 6. So what's that, like a 60-degree angle that they're going to come off at, so that every single thick filament is going to be able to make contact with 6 thin filaments around it. Okay. It's going to make these hexagons. What this lets us do is it lets us maximize the amount of actin and myosin that we can cram into a myofiber. So we can pack more and more and more protein into a fibril, therefore into a fiber, because the more of these that we have, the more interactions between the heads of the myosin and the actin we can generate. And as you guys will see next week, each individual connection between a myosin molecule and an actin molecule is called a cross bridge. And the more cross bridges you can form, the more force you can generate. People that have bigger muscles than other people, they're stronger because they can form more cross bridges. That's not always the case, but generally, the bigger your muscle, you've got more actin and myosin, you can make more cross bridges, and it's each individual cross bridge that can generate force. So if you've got more, you can generate more force than someone else. So our goal, three-dimensionally, here, structure, imparting function, is to cram as much of that in there as we possibly can. Okay?
Like the black.
Yes. It has to be weight, right? I don't know. I probably should go look that up, but it's got to be in weight. It has to be in weight. And you can ask, is that wet weight? Is that dry weight? Or something, right? So Gabe has actually had some interactions with that. When you take biopsies and look at those things, it makes my eyes want to go crossways and reminds me of bad times when I was a grad student. Okay. Okay.
So
Yes, ma'am.
in that picture, there's the A and B.
Mm-hmm.
Should there not be two actins between each myosin? So like in the A, there's only one. But it seems like there's two between each myosin.
So all you're seeing here, they're just showing you kind of the top and the bottom. But there's one that's here and there should be another one kind of down here on the bottom and another. So you're just not, the way it's just drawn is so that you can't see all of them. So it's a good question. Okay. So I want to talk a little bit now in our last like 5 minutes there about a little bit more so about the myosin molecules themselves, because myosin is really cool. There's a lot of things that go on with muscle and function that are very much related to things that are going on in the myosin filament, right? So if you look at a myosin molecule, so here's one myosin molecule here, okay? Myosin is made up of two segments that are called meromyosin, right? You have light meromyosin and you have heavy meromyosin, okay? And so light meromyosin is going to be the tail region, right? The tail region. And so on the tail region, there's going to be these two chains that are going to be intertwined that are going to be down here, okay? And then the heavy meromyosin is going to be the head region, and then what they're kind of calling the hinges up here, which is kind of almost like the neck region or something that's going to connect these two things together. Okay. The end of every myosin molecule, the head, is going to occur twice. So these two globular heads. Okay. Not to be confused with light and heavy meromyosin. On the head, on each of the myosin heads, okay, here, what we're going to have is two myosin heavy chains and two myosin light chains. Those are different things than light and heavy meromyosin, right? So here is the head region. You've got these outside portions here. That's the myosin heavy chain, okay? And then on the inside, you've got myosin light chain part 1, and then light chain part 2 here kind of in the middle, even though it now says there's light chain part 3. It's going to be down here. What it really is that 1 and 3 are identical to each other, and 2 is a little bit different. Okay? So At the ends of the myosin head, okay, you have an enzyme called an ATPase enzyme. So it's an enzyme that splits ATP, uses ATP, okay? And then you've got the portion here on the end that is going to bind to actin, right? Myosin heavy chain here and here. The heavy chain is the thing that differs between different muscle fiber types, okay? You have fast twitch muscle and slow twitch muscle, or you have slow twitch and you have the two kinds of fast twitch, or there's really like 7 kinds of fast twitch. The thing that differs is going to be the heavy chain, okay? The heavy chain helps to regulate the function of this ATP. When you use energy, when you move, a lot of that energy that's being used is all coming from this myosin ATPase. Okay, so if you, my Apple Watch says, oh, I should have done this, I meant to do this. I could turn this on, and I could have been like, I'm going to walk around in the front of the classroom, and it says I've burned 100 calories from standing here. What that means is that I've used enough ATP primarily from my myosin ATPase to burn that number of calories. Okay? So we're going to talk a lot going forward about kind of differences in heavy chain and what that means functionally, and then some differences in the ATP. Okay? In most people, actin is the same. Okay? It's the myosin that's a little bit different. The myosin is the thing that really matters. The myosin is the thing that can change a little bit with training or disuse.
Okay, so just so I can pass the heavy neuromycin.
Yes, is this whole region.
Okay.
It's all of this together.
Okay, but then you can divide it into
Correct.
the heavy chain and the light chain.
Correct.
Got it. Okay.
The heavy neuromycin is the head and the hinge region. And then within that, in the head region, you have heavy chain and light chain.
Okay, so
That's
the
correct.
heavy neuromycin is made of like the, I
Yes.
guess the neck region, the hinge region
Yes.
is made up of the 2 heavy chains connecting.
Correct.
Okay, got it.
Correct.
Does the light chain lie near my does that have both heavy and light?
I don't know, Gabe. That's a good question. We never talk about the frickin' tail region. It's just like they're all wound together, right? And they just kind of are tangled up and they help hold the structure because they don't really regulate anything else that lets us get force or use energy. We never really talk about that. I don't know. I can go look it up. I can go try to figure it out. Okay? If you want to be a great sprinter, okay, this enzyme is the thing that makes you a great sprinter. How fast that enzyme can catalyze ATP. It's what makes you a great sprinter. Okay. This is mostly genetically determined. So if you're a great sprinter, thank your parents or curse your parents. Okay? It's much more difficult to run fast. Like we can't train how fast you can run nearly as well as we can train endurance capabilities. I can take every one of you and make you a marathon. And by make you, I mean I could train you to where you could actually run a marathon. Might not run very fast, but you could do it. Okay? Probably, I don't know that much about any of you guys in here, but I probably can't make any of you a sub 10-second sprinter in 100 meters. Probably can't make any of you a sub 13-second sprinter in the midst of all. Maybe some of you are great sprinters. I don't know. But that's an issue of I can't do very much about this. I can't make this enzyme work faster. Okay? No matter how much I train, if I train, you would actually get slower. So it was thought in the sprinting world that they train and then they rest like the month before the competition. This switches back. Anyway, that's a whole other. We'll talk about that with the fiber type stuff. Okay, but this thing here, this is what's going to determine the fiber type. It's in the myosin. Okay. All right, that's probably pretty good for today. We'll stop here. Okay. For Monday, all right. Monday, the first thing we're going to do is we're going to have a quiz. We're going to have a quiz. It may be over things that have already been, we've already covered, or things that will pop up in the rest of the neuromuscular anatomy PowerPoint. Okay? Fill out the learning objectives. And I will let you use those to take the quiz. Therefore, you should make 100. Okay? If you want to write out and reproduce every slide, great. But just know you're going to get 10 minutes for your quiz. You ought to know where things are in there. Okay? So we'll do a quiz first. Okay? And then we'll finish this off. And then your critical thinking questions for neuromuscular anatomy will be due next Wednesday. And we will hopefully, with next Monday and a little bit of Wednesday, get through this PowerPoint and we can do some critical thinking discussions. Okay? Cool. All right.